I'd like to introduce our next laureate, Dr. Pascal Mayer. He's CEO of Alphonsus and affiliated professor at the University of Strasbourg. He completed his PhD in macromolecular biophysics at the Institut Charles Sadron on the University of Strasbourg in 1991. His thesis explored the dynamics of DNA during pulse field gel electrophoresis and the creation of associated automated optical instrumentation. Importantly, we're in Canada. His first postdoc was at the University of Ottawa from 1991 to 1994, where he worked on the physics of DNA in gel sequencing. Following a second postdoc in France, he initiated his foundational work on DNA sequencing on self-forming DNA colonies at Glaxo Wellcome's Research Center in Geneva. This work continued at Serono and Mantea Predictive Medicine in Geneva until 2003. And as you've already heard, this work became a key component of today's next generation parallel sequencing. Currently, in 2014, he served as co-founder and CEO of Alphonsus, a startup company using original artificial intelligence algorithms to assemble non-pharmaceutical ingredients into safe and patentable formulas with strong biological activities of medical and non-medical interest. 2024 Canada Gairdner International Awardee Dr. Pascal Mayer will be presenting from next generation sequencing to next generation therapeutics, simplicity as a paradigm for scientific disruption. Please join me in welcoming Dr. Mayer. Disappeared? The title disappeared?
Bonjour, merci beaucoup d'être présent. Par égard à mes compatriotes et cousins québécois et acadiens, je vais commencer en français, but for the sake of being understood by the rest of the world, I will finish in English. As the introduction, as you realize, there are basically two places in the world where this technology was incepted. One you have been presented in Cambridge, and I'm going to present the roots in that I called the Lemanic roots. And Lemanic is because the Geneva Lake is also called the Lac Léman, and the area around is also called the Lemanic area. So the first thing is going back to my background. I originate from a small town in the northeast of France, a coal mining town, and I made all my studies in Strasbourg, where I met, thanks to linear algebra, my wife Mireille, who is here today, and during my thesis we had our two daughters. I started working on DNA technologies during that thesis up to a postdoc in Bordeaux again, going through Canada. And I was touching many different aspects, including instrumentation, computer programming, biology, biophysics, etc., which was the perfect profile for not finding a job, neither in academia nor in industry. So by that time, as has also already been presented, it was really very cumbersome to make sequencing, especially using gel electrophoresis or later capillary gel electrophoresis. It was slow, cumbersome, and I didn't want to spend the rest of my life making gels or running these kinds of instruments. There were some new techniques coming up, but all of these techniques had something in common, is that if you wanted to get one piece of sequence, you needed to manipulate one sample. So if you want 1,000 pieces of sequence, you needed to manipulate 1,001 million test tubes. And radioactivity was much used by that time, for those who remember these days. So while I was desperately looking for a job in summer '96, I had some time also to think a little bit. Still, doing better than gel electrophoresis, and I thought about the different things I did during my path, self-organizing structures, microbiology, PCR that I made at a journal club at some point. So it was lot of physics and Greek with on macromolecular biophysics, also single molecule observation, DNA molecule observation by video microscopy. Well, it's not a single fluorophore, there are plenty of fluorophores, but gave me the opportunity to be also, like David, confronted to very sensitive detection methods. And at some point, thinking of something, you have an idea, and then you need to find a place to put it in practice. Luckily, I was hired by Glaxo Wellcome in Geneva and arrived in November '96. And they wanted me to work on the sequencing by hybridization with radio-labeled probes, and it would have meant manipulating tons of radioactivity. And so I said, okay, guys, maybe we first pay attention to my idea. And the first scientist I was talking to was Laurent Farinelli, with whom we made the first experiments. And the picture I'm showing here is the very first picture of DNA colonies, or DNA clusters, I call them colonies, that is observed here with confocal fluorescence microscopy. They were made on a glass plate, but many things had to be done since then. So the first patents were filed April 1st, '97 by Glaxo Wellcome, and later I'll tell you why it was patented so quickly. And what are in these patents is things that we could only present briefly at a conference, which was named Automation in Mapping and Sequencing, because by that time it was all about robotizing the very cumbersome sequencing technologies. And so I presented this. Shankar left me the honor to present the very complicated, at least to explain way to make DNA colonies. It was basically you make PCR, like the PCR that we all had during the COVID, but instead of doing this in a test tube, you attach the DNA primers and the DNA fragments you want to amplify on the surface. And when you do this, in fact, you realize that different DNA fragments amplify locally and end up making what I call DNA colonies, in analogy to the way bacteria multiply on an agar plate. Then simultaneously and independently, and also in stealth mode, there was also a solid phase amplification method developed in the USA by a company named Mosaic. George Church was also involved a little bit in this. And so I need to explain very quickly what's different between what we were doing and what they were doing. Basically, when you're making PCR in solution or in the solid phase, you want to check whether a certain sequence of DNA is present in your sample. A lawyer would say you want to know if Paul is in this room. So in doing this, you have two pairs of primers that are specific of the sequence you want to amplify. You make the PCR either in a test tube or on the solid surface. And if there's an amplification, you know whether Paul is in the room, whether your COVID is in your nose, or whatever. What I wanted to do is something very different. The idea was to fragment the genome, either randomly or by restriction enzymes, and to tail them with two universal pair of primers. So each fragment of different sequence is tailed by the same pairs of primer. This allows then to amplify them on a surface with only one pair of primer on the whole surface, however large it is, and to get the DNA colonies. And so the answer is not now anymore, is Paul in the room, but the question would be, who is in the room? And I get the identity of every person in the room. That's the idea behind. So back to that famous conference, I presented the stepwise base-by-base sequencing. We later realized that the concept was first presented in a patent by Pepe Ross in Rogers Tsien, a future Nobel Prize lab, but they didn't get the funding to work on this. So it was still available, so I presented the principles and especially how we applied this to DNA colonies. By that time, we had only one color nucleotides and only on 2 of the 4 bases. So the first picture presents, try to use this, when what happens when we incorporate a C on, as Shankar presented, on DNA fragments with a certain primer on them. The first one is supposed to incorporate a C, but not the second one, and we see DNA colonies appearing. Here it's observed again by a fluorescent microscope. When we then incorporate the T, we see 2 things. One is that the colonies that were present in the first image are now no more intense, and it can be quantified. You see the numbers here is the peak intensity on each colony. And we see also other colonies appearing. So this showed that we were indeed able to incorporate specifically the 2 bases. And the third image is what happened when we incorporated the C, and nothing basically changes in the images, showing that we were indeed possible to do this. So we also wanted to sequence 10 bases or 20 bases, knowing that we were making the constructs with a restriction enzyme, which gave us more information, 6 base more information, which we can argue that it's enough to position it. And in order to make de novo sequencing, we had ways to prepare the templates such that we could, in fact, sequence fragments of a few thousand bases by bits of 25, and then you have longer reads that you could have been done. It's a long story, which is still a debate now. So I presented the performance, but by that time, my manager asked not to put the real expected throughput, only the rigorous extrapolation of what I was showing, which ended up to be 100 times faster than existing methods, but already quite more attractive in terms of cost. But we know already, like Shankar, that the throughput would be enormous, but because by just improving the density of colonies, the time of exposure, a number of things, in reality, we were totally conscious that it would be between 100,000 and 10 million bases per second, and costs around $100. So for those who buy the Illumina kits, you can have an idea. So next thing is, second part of the Lemanic route is that I convinced at some point Serono to create a spin-off in order to further develop the technology, which needed plenty of different specialties to work together. And we founded Mantea Predictive Medicine, initially Geninex. And our CEO, who was the former head of marketing of Serono, wanted to not only to make the instruments, but he said we need to make simultaneously the instruments and the personalized medicine that goes with it. And we were thinking of personal genome card to be used for pharmacogenetics. Now here are the instruments where we were on summer 2003. You recognize the microscope, and if you would look today in an Illumina machine, you would still find the basis of fluorescent microscope without the observation thing, computer like there were at that stage, pumps, valves, etc. All it takes. And here I present some of the technological details, but I go quick. And just to show where we were in the good days, so we were able to sequence up to 10 bases, and you can follow here on the image that the 2 first are not supposed to be incorporated. The first one is. We see a signal. We had also our own cleavable terminator chemistry, not as elegant and efficient as Shankar's one, but we had something that was working. At least it was just needed to have more regularity on this. So at this stage, I want to thank all these people who work beside me on getting all this done. It is a long list. You can find it on my LinkedIn profile if you want to check. And I want to stress in particularly Laurent Farinelli, who was the guy I first met at Glaxo Wellcome in '96, and who was, he was in the lab already. I barely knew where the coffee machine was. So together we made the first experiments and got the first results I showed earlier. So many things are, how did this end it? But our main shareholder, Serono cash-rich company, decided for some reasons to stop all this. They liquidated us and they sold, I guess you can admit it's valuable items to Solexa. As is written in a book, it seems that a few weeks later, you got the first sequencing results and the first VX sequencing thanks on using our instruments that I was told. And then you made a very good business that Serono passed. Okay, so what does this technology do? It has been explained, 1,000,000 times faster, cheaper. Also, this technology was used to identify the presence of coronavirus in the first COVID patients, and this is really due to the fact that you can make this anonymous sequencing of every DNA that's in a sample and not to make a hypothesis on what you are looking for. Then, I take this that way. We had several things also, but to make it short here, there's also, this technology is also used in order to find the genetic roots of disease. And this is one example, and what comes out very interestingly is that you don't get one root. There are several genes linked on strongly linked on certain disease, not talking on those that are less strongly linked. And this means that there's something wrong with a traditional pharmacological paradigm. It says you can cure somebody by having one compound going to one target and have one biological effect. Here you don't know which one to choose. So I believe that a revolution is needed in this field, and as you know, in France we are good at revolutions. So the approach I'm thinking of is can be seen as inspired by the strategy of 1,000 cuts. So what does it mean? That we want to perturbate the cellular system with massively diverse mixes of compounds. So you don't have one molecule acting on a single target, but you have plenty of molecules acting simultaneously on many targets or biological features like membrane rigidity, pH, whatever you can think of. This can look like this when you have the molecules acting. In fact, if one molecule is acting to push the system in one direction, another molecule would act to push it in the other direction, and most often the effect is neutral, but in some more rare cases you can have a strong biological effect while not having eventually deleterious effect. It's inspired from a certain number of work, and I had to publish also some work that gives the statistical biophysical roots of what I'm doing. And in order to reduce it to practice in a tractable way, we are working with mixes of edible plants. So it can, I see all the objections raising in the room, but I can assure you that while it takes more than 1 minute, in less than an hour, I can wipe all these objections out in a convincing way. So good thing is that the regulatory agency encourages us to make such experiments by encouraging us to eat very diverse foods. So there's normally no concern about toxicity on making mixes, and the theory shows that this is indeed the case. We're making water extracts for the chemists who are in pharmacy. When you have a heat, you spend 2 to 5 years in making it water-soluble. By using water extracts, we have already water-soluble molecules, and in some sort of massively parallel supramolecule phytopharmacology. When you come with a new technology, it's always difficult to describe and to give it. So it's and the very important thing is also that this is patentable in contrast to classical phytochemical approaches. Because it's synergistic, you get a composition patent mix, which is very important to later protect the discoveries and the clinical validation. There's an issue, however. If you take a library of around 100 300 to 1,000 different botanicals, and you want to make mixes of 20 of them, you have a gigantic number of possibilities that no organization can handle exhaustively. So we thought of something, of using AI specialized algorithms to do this, and these are themselves inspired from algorithms we were thinking of in Manteia in order to eventually find the genetic roots of the diseases, because by that time the geneticists were saying, no need to sequence whole genomes in days. We don't know what to do with the data. So we had also to think about ways of analyzing the data. So that's what I basically did. It goes a little bit like this. We have the plants. We have the biological assay in vitro. So it's like in classical drug screening. We make the assays and we enter the data into our algorithms. And this is since 2015, so it's not a portal to ChatGPT. And they propose us potentially improved work methods, and we circle this iteratively in order to get this done. We did this during COVID to find an anti-coronavirus. So we started with the classical experiments on Vero EC cells infected by the coronavirus. Here, each bar represents the effect of one extract, of one water extract of complex mixes of edible plants, as I call them, work maps. And most of them have no or very little effect, but we enter this data in the algorithms, and they propose us potentially improved mixes. We assay them again, and then again, and very quickly we're able to come to mixes that prevent most of the mortality of these cells in vitro. It took us a few weeks during COVID, confinement, and Christmas and New Year, so it's very, very efficient. Then we tested it on animal models. This is a model that was first, the debate was, should we use a model where all the animals are dying, or should we only use a model where only the lungs are infected? We had the model which was available where all the mice die when they are not treated. And here we got 30% survival when they are treated by oral route with our mixes. And these in reality are real survivors. We redid the experiment and show that these ones survive longer. And we see it also on the weight curve that the survivors really recovered. And we wanted to then to make a clinical evaluation of this. So we produced the product and registered it as a formal food supplement that we could also sell, and we tried it on ourselves. But unfortunately, we didn't get traction enough in 2021 to make a clinical evaluation of this. No comment. Then another aspect that's important for what we will be doing now, here we see the efficacy of our mixes relative for the reference strain with the different variants that have been tested. And in gray, it's a precision medicine, which I don't cite here. It's not chloroquine, it's another one that is seen as much more valuable. And we see that we are kept the efficacy in all the strains, whereas the precision medicine lost its efficacy already in some of the strains that were found in 2021, which is something expected with a single, 1 minute. Okay, so I have 3 on my timer. And we have also a lot of data on bacteria, on the resistance profile, which are very good. So here is an update of all the results we have already. And then we are thinking of something, the big picture really is to enable personalized medicine. That is, we would have a database of relating the compositions, the effects, and the AI. And when we have a patient diagnosed and its genetic makeup with this data, we can propose a personalized preventive and curative work map. And before making all this picture, we want to respond to the big threat today that the WHO is announcing, with millions of people dying in the coming years because of bacterial resistance. And we would use our products that are already active against these bacteria in order to have therapeutic boosters that the doctors could prescribe on top of the antibiotics they are used to prescribe, and so they would recover their efficacy on the first prescriptions and not as of today after 1, 2, 3, or 4 prescriptions, where in some cases you can have 20% mortality because of this. So that brought me to the end. So I want to thank especially the Gairdner Foundation, the associated committees, the nominators and supporters, the whole teams again that are behind all this work, and the scientific community who is using that technology and making demonstrated in some sense its importance. And I must say also to all those teachers, mentors, one of them is here today, Professor Gary Slater from University of Ottawa, friends and family, all of these people who forged me and allowed me to make this, I hope, become a reality. Thank you very much.